Hi, I'm Brad Power, and this is the Cancer Patient Lab. And we're honored today to have Wayne Carter with us. He's going to tell us about technology and a startup that he's leading that has an innovative approach to mute, um, sort of immunotherapy, an engineered immunotherapy, which is a big opportunity that a lot of folks are very interested in. Let me just handle the housekeeping items first. There are three that we just typically do at the beginning. This is for information purposes only. This is not medical advice. We try to arm patients and caregivers with information that they can take to their medical team. The second is that this is all public. Everything you say can and will be made public. So if you're concerned about that, hide your image, change your name, and don't say anything. And finally, we are a nonprofit, patient-led community, learning community, and we'll appreciate any donations that people might make, and you can do that through our website. And with that, I'll turn it over to Wayne, just to introduce Wayne briefly. As he was just saying, he's based in Athens, Georgia. It always helps me to locate where people are, but his company is headquartered in Kansas City, which is, I think, where David Plunkett is. And they've got a novel approach to an engineered immunotherapy. I was introduced to Wayne by Chris Schuler, who introduces me to somebody every week, practically, it seems, who really knows his way around brain cancer. They're starting in glioblastoma, but their technology has wider applicability to a number of cancers. So with that, I'll turn it over to you, Wayne.
Yeah, thank you so much. I really appreciate it and appreciate the attendance and obviously open to any questions that you have. I'm going to actually start with the image behind me because you may have interest in it. This is actually a T cell, and you can see this T cell is sending these long finger-like projections, and it's actually binding to the cancer cell, which is behind me, and then injecting toxins into that cancer cell and killing it. And that cancer cell is undergoing apoptosis, and that is the essence of our technology. I'm going to share slides, and again, happy to answer any questions as we go through here. I'm just going to verify that everybody can see these. Can everybody see the slides? Good. Okay. So first, Brad, I appreciate the introduction. I do want to clarify one thing. These are not engineered. This is not a genetically modified therapy. This is a personalized therapy. And so as opposed to saying it's an engineered technology, I would say instead it's personalized. It is completely personalized to the individual patient. And thus we also believe it can treat potentially any cancer because it is so personalized. That is why we have the thumbprint up here to represent that personalization. Just a disclaimer slide. And this is just a quick highlight. If you put a name to it, our technology is a polyclonal cancer neoantigen specific adoptive T cell therapy. And happy to dig into that if anybody wants to dig into that further. We do have fast track designation, which has been given to us by the FDA for newly diagnosed glioblastoma. We do believe it's a platform technology, again, because it is so personalized. And I'm going to show you some of the data which represents that the broad applicability of this. I'm not going to show you. We actually have data in stage 4 renal cell carcinoma patients. I'm not going to show that data to you today, but again, we do believe it's a platform technology. We have received a $2 million grant from the FDA Office of Orphan Products to help develop the technology. This is the way the technology works. The surgeon removes the tumor. That tumor gets sent to our manufacturing facility. There we isolate about 50 million cells from that tumor. We then attenuate the cells with 50 gray of radiation, so they're still alive, but they're no longer capable of growth or replication. We then combine those cells with GM-CSF, which is an adjuvant, and then we vaccinate the patient twice against their own cancer. And as most of you know, you generally don't have a good immune response against your cancer. And cancer has figured out how to evade our immune system. So this is really the first time that you will have a good immune response against your cancer. We also know that there have been literally hundreds of failed vaccination trials, and vaccination by itself generally is not enough to cure your cancer. We're doing vaccination in this situation, in this therapy, to prime your T cells against your cancer. We then collect those primed T cells via leukapheresis, and that leukapheresis sample gets sent again to our manufacturing facility. We activate and expand those T cells and then infuse them back into the patient, giving back generally about 50 billion activated effector T cells, and then follow that up with low-dose IL-2 injections. And so those T cells will literally circulate throughout your body and identify the cancer, just as I described at the start, and then bind to it and then kill it, and bring in other cells which are capable of killing those cancer cells. Our vein-to-vein time is about 7 days, which is really quite good. But I'm going to explain in the glioblastoma study why we actually have a greater separation between leukapheresis and the T cell infusion. So this is just an overview slide, easy for physicians to administer. We have a very favorable safety profile. These are your own T cells. They're not genetically modified. And so there are really very few side effects associated with this therapy. Next, I'm going to show you some of the clinical benefit. So we actually have a sister company, and that sister company is focused on animal health. Same technology, but it's being used for dogs. And in this case, dogs with bone cancer. And so dogs get bone cancer, osteosarcoma, just like people do. When they get it, they're usually dead within a couple of months. You can do amputation and they'll live about 4 months. You can do amputation and aggressive chemotherapy and they'll live about 10 months. In this study, we did amputation followed by the immunotherapy. And so this vertical line in this Kaplan-Meier curve really represents the median survival that you would get with amputation alone. Obviously, you can see there's a lot of survival that's occurring beyond that, and that's really the benefit of the immunotherapy. This was a 2-year study, and as you can see, half of the dogs were alive at the end of that 2-year study, and they actually went on to live many months and years thereafter. The survival of those 5 of the 10 dogs went out to almost 2,000 days, 1,900 days, 1,500 days, 1,000 days, et cetera. And so these were old dogs to start with. This was naturally occurring bone cancer. This was not a contrived model. This bone cancer developed naturally in these dogs. And so as you can see, it was really quite a significant therapeutic benefit in these dogs. And for all practical purposes, we believe that several of these dogs may actually have been cured of the bone cancer. This now, Elias is the company. They've gone on now to complete their registration study, and in January, this was actually approved by the USDA Center for Veterinary Biologics for treatment of bone cancer in dogs. Now moving on to our human work. And so this is a Kaplan-Meier curve that shows recurrent high-grade glioma. So we treated about 43 patients total with recurrent high-grade glioma. And a total of 43 patients to date have been treated. You can see here with recurrent GBM, these are mostly of stage 4. There were a few stage 3s in here. The median survival is between about 7 and 8 months. And if you look out at the 43 patients that we've treated, if you look just below the 50% function line, you can see we moved survival out to about 17 months, which is good. The most important finding, especially, is at the bottom. And so here you can see that about 20% of the patients went on to live 5 years or longer. So from a statistical perspective, obviously, it's statistically significant. It's important to appreciate these patients were treated only with the surgery. This was the second surgery, at least, after their GBM, and then the immunotherapy. So there was no chemo, no radiation therapy associated with this therapy at this point. This is one of the patients, and you can see here a very large enhancing mass. This was actually 3 months after surgery and just prior to the start of the immunotherapy. 2 months later, you can see that mass has decreased significantly in size in these 3 slices. 8 months later, you can see that mass is essentially completely gone, mainly just showing scar tissue, which is remaining. This was a long-term survivor that went to live over 5 years and longer, then they were simply lost to follow-up. In this situation, you can see literally those T cells were doing the work of a surgeon and literally debulking that tumor. we actually believe that the T cells are best designed to clean up minimal residual disease, which is left behind after a surgery, rather than actually going in and debulking a surgery or a tumor like they did here. And so that's why we've designed our current study the way we have. And so our current study is focusing on newly diagnosed patients, and specifically MGMT negative. These are the patients that do not respond to temozolomide. Their median survival is 12.7 months. Our patient population, our total patient study will be a 120 patient study, 1-to-1 randomization, with the control group receiving radiation and temozolomide. There are three key differentiators in this study in comparison to what we've done in the past that we think are going to differentiate this study in comparison to what I just showed you with the recurrent. First of all, the immune cells are going to be generated in these patients while they have a healthy immune system. In the previous study, as you can imagine, the patients, they had a very suppressed immune responses. They received, multiple rounds of radiation, chemotherapy, et cetera. These patients will have a pretty, naive, undisrupted immune system, except for aging, but certainly not affected by chemotherapy or radiation therapy. These patients will also have minimal residual disease when they have the immunotherapy, and that's going to be brought about by a combination of the surgery and the chemoradiotherapy. And then finally, the benefit of the chemoradiotherapy in this situation is it also does convert an immunosuppressive microenvironment of the tumor into a more immunostimulatory environment, which again is very favorable to the function of the T cells. So we believe that these three things are going to come together and bring about significant efficacy beyond what we've already seen before in the recurrent GBM setting. This again is just a brief overview of the inclusion criteria, pretty standard. Again, they have to be MGMT negative and newly diagnosed to qualify. We have to have sufficient tumor to manufacture the vaccine. That's generally about 1 centimeter cubed, and they have to be off of steroids. This is the way the timeline is actually set up. We do a pre-surgery informed consent. They actually sign the official informed consent after surgery, after we know for sure that they're a GBM and MGMT negative. They then receive the first vaccination, and then we continue to inject GM-CSF as the adjuvant and to continue the dendritic cell activation directly into the vaccination site for the next 3 days. Second vaccination a week later, GM-CSF, and then the leukapheresis. It's at this point that we then do the standard of care, which is radiation and temozolomide. And then after they're completed with that, after a 6-week period of time, then we give them back their healthy activated effector T cells, and really, if you will, sort of replenish their immune system, and then follow that up with low-dose IL-2. We're using a special needle for the intradermal vaccinations. It very easily facilitates intradermal vaccinations. You get a very nice response, obviously, from the immune system intradermally as opposed to sub-Q or IM. And our control group looks essentially like this. The control group will receive adjuvant temozolomide. Again, we know it really doesn't work in these patients because they're MGMT negative, but it is standard of care, so they will receive adjuvant temozolomide. The immunotherapy group will not, because obviously that temozolomide would interfere significantly with the function of the T cells. So these are the sites that we have open: USC, University of Kansas, Cedars-Sinai, Capitol Health, Piedmont, and Providence. And then we're working on opening these additional sites and others beyond this also. We're focused on GBM. It gives us our quickest path to an approval. We do believe that this has potential for potentially any cancer. Obviously, we need to have significant cancer tissue to develop the vaccine, but we do believe that it has a potential to treat many different cancers, and certainly are very excited about trying this therapy in other cancers in the future. And that's my last slide. So happy to answer any questions that anybody may have.
So as usual, I'll help moderate, and please use the raise hand feature to signal if you have a question. And Gita has one. I'm going to use my prerogative as moderator to ask the first question. Wayne, there are a lot of choices in that timeline that you laid out. the adjuvant therapies, the when you do this and when you do that. I know, is when I had my chemo, is it like 5 rounds at 3 weeks apart, or is it 6 rounds at 2 weeks apart, or like there are a lot of variables in there. So you've got both, which elements you have in that treatment regimen, and then the respective timing. How have you arrived at that? Because there were a lot of choices embedded in that.
Yeah, so it's a good question. I'll back up to it just so you can see that sort of timing right there. And I mean, it obviously we worked with experts, and actually many different neurosurgeons and neuro-oncologist experts that really helped us in the design of this. We recognize that especially with the recurrent, again, we were with the neurosurgeons and neuro-oncologists, we were working a lot with Dr. Andy Sloan, and also Mike Shalitz and Dr. Camerata. And we certainly had efficacy and good efficacy, but we recognized that we were fighting this issue of, how can we treat patients when their immune system is as healthy as possible, because that's what we're relying on here. We are relying on the patient's own immune system to treat their cancer. And so obviously we have designed this and designed all of the vaccination and the collection of those T cells up front before the patient ever receives chemotherapy or radiation therapy, because it does have a significant detrimental effect on the function of your immune system, just as steroids do also. These patients, they're all on steroids after the surgery because you need to reduce the brain swelling. But we are very careful to make sure that they're off those steroids before they then can go on to the vaccination protocol. And the same with the T cell infusion. They've got to be off of steroids before they receive the T cells, because again, we know that those steroids can have certainly a negative effect on the T cells, and we want to make sure that the T cells have their best opportunity as possible to identify and then kill the cancer cells.
Could you speak to the last section there, the IL-2? So it happens that I just had leukapheresis a week ago. I could show you the catheter hole in my neck. And I'm going to be getting CAR-T in the middle of September, but it will not have an adjuvant like an IL-2. How did you arrive at that, and what's the significance of that?
Yeah, so again, it's derived from experts that we've been in consultation with, and the IL-2, it's a low dose IL-2. First of all, you don't have the clinical side effects that you have with it, but you do get the continued expansion of your T cells in vivo, and so that's why we're giving it low dose, basically every other day for a total of 10 days, and it's just to continue that in vivo expansion of your T cells.
Thank you.
And there are some T cell therapies, and I believe TIL therapy does generally recommend IL-2 just as we are.
Okay. I'll go first with Rob and then Geeta. Rob, you have your hand raised.
Thanks, Brad. I'm here as eyes and ears for a friend of mine whose husband has glioblastoma. He had his initial surgery in December of last year and has been trying multiple different avenues with marginal success. That would eliminate basically him from something along these lines based on this tumor already being excised.
Yeah, that is correct. We have treated emergency authorization, use patients, and so that could be a possibility for him down the road. But we do have to have at least a centimeter cubed of tissue.
Now, there has been some recent growth with it, but I'm not sure whether another surgery would be in there, advisable. Okay, that's really what I was,
Yeah.
I love what you're doing. It makes wonderful sense. So.
Yeah, and just to add my commentary, it does. I'm an immunologist actually. I'm actually a veterinarian by training, but also an immunologist, and it does just make so much sense. And so, we're really just helping your immune system, educating your immune system so it recognizes your own tumor and then it can eliminate it. So
yeah, and I work on the front end of that by using nutrition and supplements to build the immune system. So something like we were working on with something that I'm working on, work put together, I think
Yeah.
we'd have remarkably
Yeah,
successful
no, I
results.
could not agree more. And so much you are what you eat, and so
Correct.
much of our diets, are contributing to
Mm-hmm.
to our cancer and predilections to
Absolutely.
cancer and so
Mm-hmm.
forth. And so that is an extremely, critical aspect
I
of
agree.
therapy, and maintaining a healthy immune system, and maintaining an immune system that's able to eliminate your cancer.
Mm-hmm. Yep, I agree. Well, thank you very much, Doctor. I appreciate your. It's a fascinating presentation. Thank you.
Thanks, Rob.
Okay, Geeta.
So first of all, Vane, thank you for what you're doing. Amazing results. Very hard cancer to treat. So I'm just curious if you are using the T cell response to select the neoantigen, or you analyze the neoantigen to select the T cell. So
No, it's a very good question.
yeah,
So we, a lot of people in, institutional investors and others will say, well, why aren't you selecting, for this antigen or that antigen or these 5 antigens? And the bottom line is, every tumor is different.
yeah.
And so we don't really, I mean, if you go back to the name of the treatment that I started, it's polyclonal. We don't care what the antigens are. We basically take all of the antigens which are presented on that cancer cell and develop them into a vaccine. And so then you're vaccinated against all of the antigens which are present on those cancer cells. And so your T cells are then being primed against all of the antigens. We're not selecting for specific antigens and then focusing on T cells that are responsive only to those antigens. We are literally creating a polyclonal response with the vaccination and then with the T cells also.
And the way you're doing that is like taking the fresh tumor tissue and then mincing it and uh, exposing the T cells that you harvested
Yeah, so
to, yeah.
the the tissue, the cancer tissue is minced and degraded, but we're actually our goal is to end up with a single cell suspension. We know that the intact cell, live cells, are better recognized by the immune system, and so we use the whole cell that's been attenuated with the 50 gray radiation, and then combined with the adjuvant. And so it's the whole cell generally, which is what we're aiming for to then vaccinate the patient against.
Okay, so the patient gets uh, after the aliphoresis, the T cells and the minced or the tumor cells get re-injected into the patient with an adjuvant. Or I'm just trying to figure it out.
Yeah, so I'm gonna back up here to
Yeah.
one of the first slides just to walk you through that again. Okay, so so again, surgeon removes a tumor, and then that tumor gets sent to our manufacturing facility. We basically use a combination of mechanical and enzymatic digestion of the tumor so that we end up with a single cell suspension. And our minimum number of cells that we need is generally 50 million. And so it's those 50 million cells or more, which are then attenuated with 50 gray radiation. And then we combine that with GM-CSF as an adjuvant. And then the patient gets vaccinated against their own cancer, live cells, but they're attenuated, so they're no longer capable of growth. So when we inject them as a vaccine, they're not going to grow, but the immune system picks them up, recognizes that they're foreign, and then develops basically you develop T cells which are now primed against those antigens which are present on the tumor cell.
Okay. I may want to make an introduction to a company that is also enhancing vaccines. So it's more like an individualized adjuvant. That's probably the best way to put it.
Uh-huh.
Yeah. But I think this is super interesting. So it's just you just need the fresh tumors tissue in order to do it.
Correct. Yep.
Okay, we have to teach all the treating physicians out there to store some at some point, because it seems to be a limiting factor in a lot of the work here. All right, thank you.
Thanks, Gita.
I don't know if you just said this, but is that fresh tissue then? And does it need to get to your lab within 24 hours, that kind of thing?
Yeah, correct. So we need fresh tissue, and generally we receive it within 24 to 48 hours. And it's just shipped on wet ice, 4 degrees centigrade. It takes us about 48 hours to turn the vaccine around, so we can manufacture the vaccine pretty quickly, and then we just store it at -80 before it's ready to be administered.
Very cool. Next up is Richard Anders, Mass Medical Angels.
Just a few questions, mostly quick. What do you think you'll be charging for this when it goes to market?
Yeah, so it's a little, early to say definitively, but I can tell you that we do think we're going to come in significantly under the price of the existing CAR Ts and TIL therapies. CAR Ts, right now are between 350 and 430,000 per treatment. TIL treatment, last I heard, I think they're aiming for 515,000 per treatment. We are planning to come in less than either of those. But we think we're going to be extremely competitive and comparable in cost, even to some of the other, PD-1, PD-L1 inhibitors, which can be as much as 200,000 per year, and you have to be on continuously. Again, this is a single treatment, but we do believe that we'll be able to price very competitively, and part of our reasoning behind this is we believe that this therapy should ultimately be available to a lot of people, and we think pricing it in this range will make it available ultimately to a lot of people.
And so your cost of goods won't be that high, it sounds like.
There will obviously, there's significant cost of goods when you have to think about this kind of GMP manufacturing, but there certainly is plenty of space for profitability of the company, and yet also developing something that we think is very reasonable for payers to consider.
A question about adverse events. I mean, you're doing significant immunostimulation with all sorts of cells. Are you seeing any autoimmune disease manifestations in your patients?
Yeah, we've never seen any autoimmune manifestations at all. Again, these are your own T cells, and so they're just T cells which have been primed against your cancer, and so there's really no reason why there should be any autoimmune activation, and we haven't seen any. About a third of the patients will have sort of a low-grade flu-like symptom when they get their T cells back, and that's about it. And that's really just a manifestation primarily of these T cells are producing TNF alpha, and so so the patients are getting a little bit of TNF alpha when they get their T cells back, and so they'll get sometimes a low-grade fever, a little bit of malaise for 24 to 48 hours, and that's really about it.
I mean, because you're injecting cancer cells, but they're not just cancer cells; they're cancer cells with normal antigens.
Yep,
I mean, there's all sorts of stuff, but it just wow, that's impressive.
That's where our immune system works the way it is, right? With the HLA, proteins and so forth. So you recognize self, and once you recognize self, then you don't develop an immune response against it. So yeah, we haven't seen any autoimmune.
Wow, that's impressive. And just a sort of an in the weeds question, doesn't really mean anything except for curiosity. It looks like you were a 1-to-1 randomized clinical trial.
Correct.
I'm just wondering why you didn't do a 2-for-1 or something. The effects of glioblastoma are so incredibly severe. You'd get signal with a 2-for-1 randomization, or probably a 3-for-1, and you'd get more patients in the trial. I'm just curious, but not a big deal question.
Yeah, so we've had that discussion, and with the FDA, with investigators, and there have been, as you can imagine, a lot of different opinions about the right way to do it. And ultimately, I think, I guess we sided with the FDA.
FDA wanted 1-for-1?
Yeah.
Because I imagine patients would have loved a 2-for-1 or 3-for-1.
Absolutely. And, you know,
Yeah.
we've also considered a synthetic, control arm, all sorts of things. But right now we're just focused on 1-to-1. And so, yeah.
And it's not hard to recruit.
Well, it's been slow. There's no doubt about it. You know, our recruitment is slower than what I want, but we're moving forward. So,
Wow, it's neat stuff.
yeah, thank you.
Let me just handle a couple of questions on the chat before I go to Mark Stoner. Bill Passman said, I noted that your paper lists 2000 as the image date. How long have you been doing this?
So that's honestly the biggest frustration I'd say that the company has had. And quite honestly, the founder of this technology, Gary Wood, I would say that he was well ahead of his time. He developed this technology and started treating patients in the 1990s. And so some of the patients the recurrent patients that some of the ones that were on that graph were treated back in the 1990s. By the time, so the company was actually founded in 2006. And by the time the company was founded, immunotherapy, PD-1, PD-L1 inhibitors were beginning to take off. Nobody was interested in T cell therapies or cell therapies in general. And it really wasn't until CAR Ts were approved that there began to really be a significant interest in funding T cell therapies. And so we struggled so much with funding on the human side that in 2012, the company tried to do an IPO and there simply wasn't the support. And so pulled back. And that's when we shifted to animal health. Kansas City is known as the animal health corridor of the world. We were able to get investors to develop it on the animal health side. And as you can see, we now have an approval for dogs to treat bone cancer way ahead of treating anything in humans. And it's simply because the funding wasn't there. And so that is the reality of technology and the funding environment. Now, obviously, we've been able to get fast track. We have an FDA grant. We have investors that are interested in moving this forward now. And so it is certainly a different environment. It's still a challenge. It's still a significant challenge to raise the money that's needed to do this study. But it simply was all an issue of funding and the marketplace not being ready to fund a T cell therapy.
Thank you. Picking up on your comment about immune checkpoint inhibitors, Roger Royce asks, do you combine this vaccine with checkpoint inhibitors? I guess not, because you didn't list it, but have you considered it?
We've absolutely considered it, and I think that's a very logical. I mean, there are a lot of things which are very logical, and so yeah, certainly that is one of the things. You certainly could combine the vaccination with a checkpoint inhibitor. You certainly could use the checkpoint inhibitor after you receive your T cells back. So I think both are very good suggestions. The other thing, and I didn't mention it in our slides, we do have a joint venture with another company called Genelux, and they are an oncolytic virus. We believe that there's a great synergistic opportunity between an oncolytic virus and our therapy because the oncolytic virus in its action will create inflammation in the lysis of that tumor cell and bringing in other inflammatory cells. And so again, we think there's a great opportunity for synergy between an oncolytic virus and our therapy. And we actually were granted a patent specifically to give us that opportunity.
Okay, Mark Stoner, you're next.
Yeah, that was a very interesting presentation. So like Brad, I have diffuse B-cell lymphoma. I start CAR T treatment September 2nd, bridging technologies next week. Question, what's the difference between T-cell and B-cell? And also, is the B-cell a separate expansion for you or apples to oranges?
So they're obviously completely different immune cells. Your B cells are a cell which is producing antibodies. Your T cells are a cell which does provide long-term memory, immunologic memory to your immune system. They both provide that long-term immunologic memory, but one is through the production of antibodies, and T cells are simply through their ability to either directly kill another cell or bring in other cells into that, inflammatory milieu. So they are very different cells. We are only focused on T cells, and obviously using T cells ultimately to kill a cancer cell. We have generally learned, I think, through many years of immunology research, that it's really not your B cells that produce cancer immunity, and so the cells which are really important to be able to fight and potentially cure cancer are your T cells and not a B cell. Does that answer your question?
Yeah, no, thank you.
Yep.
Okay, picking up a couple more questions from the chat. Alan Morris, who's a pathologist and an advanced prostate cancer patient, asks, have you ever heard of the concept of antigen competition? And if so, what is your notion of it?
You mean as it relates to what we're doing in this situation? I guess I'm a little bit lost as far as exactly the purpose of the question.
I guess you were talking about it being polyclonal, and I guess polyclonal is sort of like there are multiple antigens that could be stimulating things. I guess could they be competing amongst themselves?
So I think there's always that possibility. I think my bias would be that I rely on the immune system, our innate immune system, to be able to figure out and maybe Dr. Morris is going to provide more insight there on that question. Anyway, I believe that our immune system has the ability to ultimately develop those appropriate responses to individual antigens, and then figure out the appropriate responsiveness there. I think sometimes I think when we try to engineer something too much, sometimes we're successful at doing that. Sometimes we can interfere, I think, with the proper function of a system.
Okay. Alan, are you off the phone? Are you able to clarify whether we understood your question correctly?
Honestly, I did not hear anything. I wasn't in the beginning of this, so it's probably going to be obstructionist of me to even go into it. Dr. Carter, antigen competition is a profound concept, and I believe it's not on the radar screen of any vaccine person. I'll ask another one. There is a thing that is on the radar screen in the basic science of molecular biology, and that's called T cell hierarchies. Have you ever heard of that?
Yeah.
You have heard of that?
Sure.
Do you believe what you're doing is a non selective way of trying to punch through that, in the sense that your process is not going to be active at punching through that, or do you think it will?
I think it will.
Okay.
Yeah, I do believe that we'll be able to overcome potential hierarchical issues that may exist within T cells. Obviously there's so much that we still have yet to learn, and to some degree we're still in our infancy, right? But I think that's one of the things that I like about this technology, is that we are relying on the immune system itself to be able to figure out how to overcome these issues of the myriad of different T regulatory issues that the cancer cells, the cancer environment, the microenvironment throws at the body. And I do believe that this is one way in which we're able to overcome that. Some of this is mass action. Obviously, I do believe that there's an opportunity to combine this therapy with other therapies, as we mentioned, the checkpoint inhibitors and others. This is not, certainly I don't believe that this is going to be the panacea treatment for everything, right? I think the combination therapy, they always tend to work better. If you put, 2 or 3 different therapies together, they tend to work better than a single one alone. But I do think that this T cell approach is moving in the right direction to get your own immune system to fight your cancer.
There's a question here from Martin Luzbatak about do you ever have a problem getting enough T cells through the apheresis?
We have not. We actually do generally 2 leukaphereses, but we have never had a problem of not having enough T cells. We also, as I mentioned in one slide, well, it's right here, our vein-to-vein time is 7 days. We could certainly expand that, the expansion time in the lab. We don't need to. Within that 7-day period of time, we're able to activate the cells and have a large number of T cells that we then administer back to the patient. We have not reached really a toxicity level on administering the T cells. And it's, I think there are many ways this therapy can be potentially expanded. We've already talked about adding the PD-1, PD-L1 inhibitors. We could potentially add in additional doses of T cells down the road. You could, there are many ways that this therapy could potentially be expanded. Our focus right now is just to get it approved and then begin to turn it over into the hands of different oncologists to try to figure out other ways to expand it. But our goal right now is laser focused on an approval.
Roger Royce asks a question here. How often do you get an immune response from the vaccine? Is it 100%? And I know this, just to add a little color to this, I know Roger has gotten a personalized vaccine recently, and he just told me today that he got an ELISpot test. So it also raises a question, are there companion diagnostics that you can use through this process that are measuring the effectiveness of your vaccine?
Yeah, no, it's a good question. So we use the DTH response, the delayed type hypersensitivity, response, same as like the tuberculin test when you get a TB test. And after the first vaccine, we generally have about 90% of the patients demonstrating a positive DTH response. After your second vaccine, it's essentially 100%. And that's across a myriad of different types of tumors also. I think there are some people that suggest that some tumors are more immunogenic than others. We have not found that. We really have seen a positive immune response really against any tumors at all.
Well, it's obviously very encouraging, given that there are a lot of cold, solid tumors that don't have an immunotherapy working. Brian has a question.
Yes. Do you do any pre or post immune profiling of the patients?
Pre, no. I mean, we're literally the only inclusion criteria for these patients in our study are their lymphocyte count. We really want a lymphocyte count, I think, of at least 800. But other than that, no, we're not doing any specific immune profiling to look at specific T cell populations or anything like that. We certainly do that post hoc, and we have a variety of different biomarker studies that we're going to be running as part of this, but not as an inclusion criteria.
I obviously what I'm trying to get at is just understanding who's going to respond and who's not going to respond.
Yeah, so are we.
If you get a better immune profile, you're going to understand who's a good candidate.
I completely agree. Again, we're sort of laser focused on getting an approval. But certainly as part of this study, we are collecting all sorts of different samples, which will be used, for immunophenotyping, for genomic analysis, for proteomic analysis. So we have a large number of different biomarker collections which are being done and which will be analyzed specifically to look at that. Why are some patients responding and others are not?
Great. Thank you.
Vanessa Hugo, I don't know if you mind me putting you on the spot, but I would really like to get a perspective of a brain cancer patient. And of course, you're the caregiver to your husband, Michael. How does this, and I know that Al Musella walked us through, 10 different possible therapies that he would recommend people consider. I assume this is on the list, but how do you rate this amongst the other treatment options that you and Michael are considering? She says she can't unmute. Okay. All right. Maybe Vanessa, if you could respond in the chat then, if you don't mind, because you're one of the few brain cancer patients we've got, and I'd just like to figure out how this stacks up in the risk return, because it seems like it has a lot of potential. Yeah, there's a request here for your email address, Wayne. We'll share that.
Yeah, please.
We'll share that around. Okay, I think we're running out of questions. There was a discussion. Roger, would you like to say anything? I know you've got a particular perspective on all this, given your work in immunotherapies for yourself.
Well, my perspective is as the patient, of course, and I'm just kind of blown away looking at this, because you've got this down to such a clear and fast process, because it took me a good 6 months to get through this here. And I did something similar to this here and in Japan. Japan, it took about 6 weeks because they don't have an FDA, but still the 7 days vein to vein. And I think you said 6 weeks to actually get the vaccine manufactured, if I designed it.
48 hours, 2 days.
Yeah, that part, that's pretty amazing.
Yeah.
Because, I know, so I had a neoantigen peptide vaccine. I also had NK and dendritic cell expansion at the same time, and some other things. And I know through Julia Leandro Foundation with the JLF, with their vaccine, just the time people sat around waiting, about half the patients died waiting for the design and testing to be completed.
Yeah.
So that's a big deal if you can, to actually get this to the patient that quickly.
Yeah, I mean, the issue, a lot of those vaccines are being, developed off of genomic sequences, right? And so that's where, all of the time gets added in. Our issue is we've got to have fresh tissue, and we need basically about 1 centimeter cubed. And so those are the limitations. But assuming that we can have that fresh tissue and enough to be able to harvest at least 50 million cells, then we're good to go.
Vanessa had a response which you may want to respond to, but again, she's very knowledgeable about all of the possible therapies, and she says this is a great therapy candidate, just limited access due to the pre-surgery trial enrollment requirement. Again, that fresh tissue and not having been other treatments. Would love to see this combined with Optune plus maybe checkpoint inhibitor. Any comments to that, Wayne?
So we've had, some neurosurgeons that have been interested in potentially adding this into Optune, and with Optune also, obviously from a FDA perspective, it needs to be as clean as possible. So we're certainly interested in potentially doing something like that down the road, just as we would be with a PD-1, PD-L1 inhibitor. But right now we're just focused on the approval at this point. So we're again, if somebody wanted to participate as an experimental use patient and wanted to use Optune in addition to this, and they had a PI that was willing to to function for them as their investigator, then we're happy to entertain possibilities there. But our study needs to be clean for the FDA.
Got it. Dr. Chris Apfel has his hand raised for a question.
Well, first of all, congratulations to this very impressive technology that you have developed and worked on it for so long. As an entrepreneur myself, I know how long it takes actually to get to market adoption, and so congratulations to this tenacity and sticking with it. I think it's very clear that everybody who is here on the call is very impressed with what you and TVEX and the founders have actually built there. Very impressive. I have one technical question, and that is in regards to the vaccination. It does look to me like after you have isolated the tumor cells after the surgery, they are injected twice. The question is why twice? The other question is actually, you said at least 50 million cells. Do you really do you know how do you know how many cells to inject? Because you also don't want to induce any T cell exhaustion. And so I am concerned that you may actually even overwhelm the immune system and the T cells, so that you actually may not actually have enough selectivity for the T cell activation and expansion. That's one question. The second question is, with the vaccination, you hope that you actually have these T cells, and they can be polyclonal because also the antigens and even the tumor itself may be heterogeneous. You would hope you have a higher proportion of T cells that are really directed against the tumor. But it does seem like the T cell activation and expansion is not during the activation and expansion. Are you exposing these T cells also to the primary cancer cells as well? So this is the technical question.
Okay, so I'm going to focus on the last question, I think, first. So you're asking if we're using the cancer cells as part of the activation?
Yes.
And we are not. We're using CD3.
And why is that?
Because you don't need to have, because you get basically a pan response to activate the cells. You get a much stronger response than actually you get when you use an antigen like a cancer antigen. So we actually get a more robust response and a more reproducible response by using anti-CD3. So that's what we use. And
then the first question is the selectivity with the vaccination. How do you know that you're not overwhelming the immune system?
Yeah, so I certainly don't believe that we're getting a good immune response. I know some of the early studies from my founder, he did quite a bit of work trying to identify the right number of cells to ultimately come up with that vaccination number that we have. We actually vaccinate the patient in 4 different locations, 2 in the axillary area, 2 in the or the inguinal area, just to sort of cover a large number of lymphatic drainage and lymph node locations. And then they receive that vaccination again twice. In some situations in the past, we've actually done 3 vaccinations. And really have found no significant difference there. So we've basically have backed off to just doing the 2 vaccinations at this point.
Okay, and I have actually two more questions. We have 9:58 on my clock here. So I try to be concise. The first one is, if I had a glioblastoma and I wanted to have it today, is there, according to the Right to Try Act, is it possible to get your treatment outside of the study?
Yeah, I think it would be possible. Again, you would essentially just be I think we would qualify you just as an emergency use authorization patient. So yeah, that would be a possibility.
And I could imagine, I don't know whether enrollment is difficult. In my mind, enrollment should be very easy, even if it's 1-to-1. And actually, from the total number of patients you need to show a statistical difference, the 1-to-1 is the ideal randomization proportion.
Right. Yeah.
So that you get with a minimal number of patients to have basically the maximum power in proportion to the number of patients exposed or participating in the trial.
Absolutely true. That's correct.
But one of my questions is, if I don't know whether the randomization is a barrier for your recruitment or whether it's not, right?
It's not. No. So I mean, there have been a variety of issues. One of the things that we recognize is that most of these patients come in through the emergency department, through the ED. And sometimes, we have to have fresh tissue. And so if a surgeon cuts on a patient before they really recognize that there's a clinical study ongoing, if let's say the neuro-oncologist is actually the PI and hasn't adequately communicated with the neurosurgeon, then we could lose a patient there. We've lost some patients because they just have done so poorly after the craniotomy, they've gone right into hospice. We've certainly lost patients because they're MGMT positive. But yeah, I mean, it's like any clinical study. They always take longer than you want them to. I'm with you. I just can't imagine why, we're not blasting through our enrollment. But bottom line is it's tough. It's why we have 6 sites open, but it's why we're going to be opening at least another 6 or 9 sites, over the next 6 months, because we need more sites, we need more neurosurgeons, and unfortunately, we need, patients, but we also want to find a solution for them.
Okay. The final question is, in the Kaplan-Meier curves, there were, what's really impressive is a 5-year long-term survival of benefits of at least 15, if almost 20%. That's what it looked on the Kaplan-Meier curve. Very impressive. What about the other 75 or 80%? It can't be just the immunosuppression through the temozolomide. Do you believe that some of them may just not be accessible through this immunological approach?
Yeah, so obviously we showed a benefit in pretty much everybody. That whole Kaplan-Meier curve was shifted to the right, and so there was a benefit in a large number of those patients. But it is important to appreciate that we are relying on the patient's immune system to fight the cancer, right? And so those patients, a lot of them are older. In this situation, they were recurrent, so they'd had multiple craniotomies. They certainly had radiation and chemotherapy to suppress their immune system. They'd had many doses of steroids, so they had so many things going against their immune systems, right? And yet that's exactly what we need to fight the cancer. It is the reality also of where we are today in our treatment of cancer. We give all of these therapies that have this negative impact on the immune system, when the one thing that your body has to potentially fight the cancer itself is your immune system. And so, I do believe that in the future, in years and decades to come, we're going to realize that the more we can maintain the integrity of the immune system, the better off that patients are going to be as far as having the ability to fight their own cancer.
Okay, just to wrap up, 2 quick questions in the chat, Wayne, and then I'll let you go. What is the dose of IL-2? From Martin Muspatak.
Yeah, I don't want to misquote, so I'd have to, but I'm happy to reply. Certainly they can email me, and I'm happy to give the dose. It is a low dose. Okay, and then Ryan Moon asks, I know you mentioned glioblastoma, but I was wondering if you're working with another grade 4 glioma, namely diffuse intrinsic pontine glioma. Yeah, so we are not, but we have obviously intentions to look at a large number of cancers, assuming we're able to get this approved or have significant funding. We'd love to refocus this again on recurrent. There's so many patients with recurrent GBM that we'd love to go after recurrent GBM, but certainly MGMT positive. You obviously you can go down the list. These patients need a therapy that works for them, and so we are very anxious to try to move into other tumor types. Well, in a quick PS, there was a question here about what would you need to do to treat kidney cancer from Bill Passman. So it's when we first did our kidney cancer study, we had a very nice improvement in survival in those patients, and that was before the checkpoint inhibitors were approved. And so today, I think it would make a lot of sense to combine our therapy with a checkpoint inhibitor and look at the benefit. So that would be my approach.